Today we're gonna learn about the DNA ligase enzyme, that how it's used to join two strands of the DNA, which is clipped. While using the restriction enzyme like BAMH1, which results in a cut-end in the DNA, and the phosphate group on the pipette region on both the sides. Well, that phosphate is required by this enzyme to join two strands. But how it helps join? Well, the first thing is that the DNA ligase enzyme won't act alone. It must be bind to ATP or NAD. Once it attaches with them, further process leads to the formation of your ligase AMP complex. This is what we call this ligase AMP activated enzyme. Once it's done, it will ready to transfer that monophosphate group. Once it goes and attach with that of your 5-phosphate region of that cut end, it's gonna transfer this monophosphate to that region. Once this bond forms between them, the ligase function begins. You can see here this energy which get clipped by this enzyme releases energy being utilized to make another new bond, and the phosphodiester bond that seals that nick the DNA. So this region get joined with this enzyme that cause this phosphate group to release, and the energy which get produced during this process will help to join these fragments together. In this case, we are using fragments of that your target gene and that is your vector. But to prevent the self joining of the two regions in the vector, we must need to remove the phosphate group. Once this phosphate group removed, it will not be get ligated by this enzyme. So once we have our target gene without undergoes this alkaline phosphatase, retaining its phosphate group, in presence of this vector which has removed this phosphate group, this ligase will be going to be helpful to join those two current completely, and that's only the case where it will be possible. Thank you very much.